in the body, there are calcium receptors in many different cell types, but there's only one calcium receptor gene. And so the same gene and the same protein has many varied and different functions in different cells throughout the body. And so one of the things that I find interesting is how can the same protein work in so many different ways to elicit so many different functions. And so, for example, some cells can be depolarized. They're electrically excitable, like neurons and neuroendocrine cells, where an electrical change in the membrane can make calcium flood in, whereas other cells are not electrically excitable, and therefore their calcium gets released from stores inside the cell. And so therefore the calcium receptor is going to work differently in those two cells. And there's lots of other examples of this. And so one of my big questions is, because we work in our own system and we study the receptor in a single cell, it's trying to compare different systems side by side to see what can we learn, what can we knowing from one system apply to another, but equally where are the really big differences in how the receptor works in different cell types. And this may have profound significance, of course, in drug discovery, because a drug may act very differently on the same protein in different cell types. And if you know and understand that, you might be able to select a therapeutic good pathway over a bad side effect pathway, and improve drugs. We may be able to develop better drugs by being able to discriminate between the same receptor working in different cell types.